So this is chapter 3, part 3. We're moving on to the cell envelope. Um, the cell envelope is the part of the cell, think of it as the coat of the cell. It lies outside of the cytoplasm, outside of the internal parts of the cell. It's composed of 2 or 3 basic layers. Depending on the type of cell, it'll be 2, and other types of bacterial cells have 3 layers. They each perform a distinct function, but each of these layers actually do work together as a protective unit. So these parts are the cell wall, the cytoplasmic membrane, and the outer membrane. So keep in mind that cytoplasmic membrane could be referred to as cell membrane. Those terms are interchangeable. Cell membrane, cytoplasmic membrane, and I think there's like 4 other words to use to describe cytoplasmic membrane, but they're escaping me at the moment. One thing that I'll say first and foremost is that when we talk about a cell wall, sometimes it's a slip of a tongue to say cell wall and we're talking about some human cells, and that's incorrect. Human cells do not have cell walls. Bacterial cells have cell walls. So make sure that you are careful when you refer to the cell wall term and what you're specifically talking about. And not all bacteria have an outer membrane, but we're gonna look at an example here. So we've talked a little bit about the Gram stain, and the Gram stain is a test that we do in the lab to separate bacteria into 2 separate categories. So instead of having every possible bacteria in the world causing an infection, we have split that pile of bacteria in half to be able to distinguish between gram positive and gram negative when we use the Gram stain. And the Gram stain is basically, you put the bacteria on a cell, you put a series of chemicals and dyes on that bacteria, and as a result, the cell will be either gram positive or gram negative. Now, in this picture, it shows the specific part of the bacteria, the bacterial cell wall that causes the cell to be gram positive or gram negative. So I'm going to talk a little bit about this, and we're going to circle back to Gram stains in a little bit, but I want to just point some things out. So first of all, these, for some reason, these totally remind me of Kit Kats or something, some sort of one of those wafers. I don't know why. It's really not anything like that, but for some reason in my head, that's what I've always thought of with these. But you can see this layer here should look familiar. Looks just like the makeup of human cell membranes. Okay, so this is the cell membrane of a gram positive cell. And, it's got that lipid bilayer, and, the arrangement is that the fatty acid tails are in the middle and the polar heads on the outsides. So it's very similar to what we've experienced so far with anatomy. On top of that is a thick, if this is peanut butter, right? And then we slap on this slathering thick layer of jelly. Okay, look at how thick that is. And if you can see this, I don't know how big this is on your screen, but in your book, there's a picture of this. You can see it says peptidoglycan. And that peptidoglycan layer in gram positive cells is really, really thick. In addition, the gram positive cells have these lipoteichoic acid molecules sticking off as well. And we'll talk about what that means for the cell later. But it's important to remember that the peptidoglycan layer is super, super thick and gram positive. In a nutshell, because it's so thick, it's going to hold on to the stain that we apply to the cell and therefore remain purple. And again, you don't have a lot of context to that statement, but as we talk more about this, and if you re-listen to this, then that'll make a little bit more sense. So when we go over to this guy, you see you have the cell membrane, and then we have this peptidoglycan layer here, which is much thinner. It's more delicate, more dainty, if you will. And then this layer, another layer of, or another bilayer, right? Another lipid bilayer. And this is on the gram-negative cells. So you can imagine if you have a really thick sponge soaking up a bunch of stain, then it's going to be hard to wring that stain out of that thick sponge. But if you have a really thin sponge and you soak up stain, it's going to be really easy to wring out that stain. And that's kind of the same idea here. In addition, when we're talking about gram stains, one of the steps in a gram stain is that we have to add alcohol decolorizer to, we apply that to the cell or to the slide. And when that happens, there's no lipid bilayer up here to be destroyed. And this is so thick that alcohol decolorizer really doesn't affect it. But over here, that alcohol decolorizer basically beats up and tears up this layer. And when that happens, the alcohol decolorizer can get to this thin layer and it takes away some of that stain, leaving the cell pink in the end, because the last application in a gram stain is the application of a pink dye. So this one was initially stained purple, and then when we add pink to that, it doesn't really change it. This one was initially stained purple, it was decolorized, and then we added the pink stain, and it kept that pink stain because we didn't wring it out again, if you will. So big difference between gram positive and gram negative is that the thickness of the peptidoglycan layer and the ability to hold on to the dye that's applied in a gram stain. Over here on the gram negatives, we also have the lipopolysaccharides and what is the other thing? To revert to my notes, it's just the lipopolysaccharides that we're really concerned with here. I know that the PowerPoint talks about porine proteins, but I want you to kind of tuck lipopolysaccharides into your memory banks for now, because we're going to talk about those later on when we talk about actual infections. And it is important to remember that the lipopolysaccharides are associated with the gram negatives, these things sticking off the cell, and the teichoic and the lipoteichoic acids are sticking off, the molecules are sticking off of the gram positive. So the cell wall helps to determine the shape of the bacteria, provides strong structural support to keep the bacteria from bursting or collapsing. And certain drugs will target that cell wall, and that's great because then that disrupts its integrity and causes the cell to lyse. And since our cells do not have a cell wall, then those drugs will not affect us in such a negative way. Certain infections, whether it be fungal infections or viral infections, they're hard to treat because of how the drugs, those microbes are similar makeup to our cells. Therefore, if we're attacking those cell parts in those microbes, then it's going to be hard on our body as well. So that's why it's easier to treat microbes with a cell wall, specifically with drugs that target the cell wall. Moving on, peptidoglycan, composed of repeating frameworks of long glycan sugar chains. Not really worried about what it's made up of. It helps to provide some flexibility and support for the cell. Gram-positive cell wall, thick sheet of peptidoglycan, as I already mentioned, contains teichoic acid and lipoteichoic acid. Notice how I changed that to a different color because I'm certain that's going to be a test question. What do they do? What do those two acids do? They function in cell wall maintenance and also enlargement and contribute to the acidic charge of the cell's surface. Gram-negative cell wall, and actually I should probably change these colors because I like things to kind of stand out. So gram-positive is going to be like a dark purple, dark blue. Let me change that to dark blue. And then gram-negative is going to be pink. So remember that. So let's see, we'll change that to pink. So gram-negative cell walls are more thin sheet of peptidoglycan, and that gives the cell, the gram-negative cell, more susceptibility to lysis. As I mentioned in the description about how that outer cell membrane can be destroyed, and then that alcohol, if we're using alcohol, can get to that peptidoglycan layer. So steps in a gram stain. So I'm going to go over this, and I think then I'll start, then I'll start the next, actually no, I'll end this here so I can do the staining all together. So we'll start up with part 4 shortly.